Hello children, welcome to biology class. We are discussing the lesson Cell, the Unit of Life. Today let's discuss in detail about prokaryotic cell. Children, we know that prokaryotes are represented by bacteria, blue-green algae, mycoplasma and PPLO. PPLO, the expanded form, pleuropneumonia-like organisms. Okay. The example mycoplasma. While discussing the first class, that means introduction about this lesson, we discussed that mycoplasma is considered as the smallest living cell, isn't it? It is included in PPLO. Children, prokaryotes are generally smaller and multiply more rapidly than the eukaryotic cell. They may vary greatly in shape and size. See children, these are the shapes of bacteria. So there are 4 basic shapes of bacteria. Rod-like bacteria. This is example Bacillus anthracis, which is responsible for the disease anthrax. Then here are some spherical bacteria. This is Coccus, Streptococcus pneumoniae, causative for the disease pneumonia. Then Vibrio. This is Vibrio. This is Vibrio cholerae, that's the name of the bacteria, and which produce the disease, which cause the disease cholera. Then Spirilla, this is Helicobacter pylori. Okay, so different shaped prokaryotes are there. The organization of prokaryotic cell is fundamentally similar, even though prokaryotes exhibit a wide varieties of shapes and function. That means the fundamental structure is same in all the organism. So now let's discuss about the fundamental structure of a prokaryotic cell. Prokaryotes have a cell wall surrounded by a cell membrane. Okay, but it is except in mycoplasma. Then the fluid matrix, see here you can see a fluid matrix inside. This fluid matrix filling the cell as a cytoplasm. There is no well-defined nucleus. Genetic material is basically naked, not enveloped by nuclear membrane. Children, do you remember in class 9 we had discussed about the structure of plant cell and animal cell. So we all know that inside the nucleus there is genetic material and the nucleus is enveloped by a nuclear membrane, isn't it? But that nuclear membrane is absent in prokaryote. That means genetic material, either DNA or RNA, which is simply embedded in the cytoplasm. That's why they use the term naked. Clear? In addition to the genomic DNA, many bacteria have small circular DNA outside the genomic DNA. See? This is the genomic DNA. Outside it is called nucleoid, and outside the genomic DNA you can see small circular DNA. It is termed as plasmid. Okay, the plasmid DNA confers certain unique phenotypic characters to such bacteria. One such character is resistance to antibiotics. Okay. In higher classes, you will learn more about plasmid DNA. It is used to monitor bacterial transformation with foreign DNA. Okay, so it is used in the field of biotechnology. Children, nuclear membrane is found in eukaryotes that we know, but no organelles like the one in eukaryotes are found in prokaryotic cell except ribosomes. Okay, so now let's discuss in detail about the cell envelope. Cell envelope include 3 parts. This is a cell envelope. Cell envelope include 3 parts. Which are the 3 parts? Glycocalyx, cell wall and plasma membrane. So if the question is asked to write about the cell envelope, you have to write about the glycocalyx, cell wall and plasma membrane. The bacterial cells have a chemically complex cell envelope. The cell envelope consists of tightly bound 3-layered structure, glycocalyx, cell wall, plasma membrane. First, let's discuss about glycocalyx. It differs in composition and thickness among different bacteria. It consists of two layers. The first layer is slime layer, the thin layer, then thick capsule. Okay, which are the two layers of glycocalyx? Loose sheath, slime layer. It is followed by a thick and tough capsule. That is about the first envelope, first cell envelope, glycocalyx. Which is the next one? Glycocalyx is followed by, yes, cell wall. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. Okay, children? So, what is the speciality of the second layer? What is that? The cell wall. It determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. Then the third envelope, that is plasma membrane or cell membrane. It is semi-permeable. In nature, what do you mean by semi-permeable? It never allows all the substances to pass through it. Instead, it allows only selected substances to pass through it. That's why we are using the term semi-permeable. Clear? So, the plasma membrane is semi-permeable in nature and interacts with the outside world. Children, so if the question is asked to write about cell envelope, you have to write about glycocalyx, cell wall and plasma membrane. These are the three envelopes of prokaryotic cell. Clear, children? Then an important thing, bacteria can be classified into two groups on the basis of the differences in the cell envelope and the manner in which they respond to staining. Children, while taking the sections, you might in class 11 lots of experiments are there, but unfortunately we are not having a chance to enter to bio lab, isn't it, because of this COVID situation. But as a coloring agent we are using different types of stains, isn't it? So according to the manner they respond to the staining, we can divide the bacteria as two, as gram-positive and gram-negative bacteria. Those bacteria that take up gram stain are called gram-positive bacteria, and the others that do not take up the gram stain are called gram-negative bacteria. Clear? Again, the structure of this is plasma membrane, semi-permeable membrane. It is also called cell membrane. Then about a special structure, a special membranous structure is mesosome. See children, here you can see mesosome. I will show you one more diagram. Yeah, it is very clear here. This is mesosome. It's a special membranous structure, mesosome, which is formed by the extension of plasma membrane into the cell. These extensions are in the form of vesicles, tubules and lamellae. And what is their function, children? They help in cell wall formation, DNA replication and distribution to daughter cells. That is the major function. They also help in respiration, secretion processes to increase the surface area of the plasma membrane and enzymatic contents. Here you can see the mesosome. Is it clear? Children, here also the mesosome. You can see the mesosome in the form of vesicle, isn't it? Then, in some prokaryote like cyanobacteria and all, there are other membranous extension into the cytoplasm called chromophores, which contain pigments. So, for this formation of color, pigment means which give color, isn't it? So, rather than this mesosome, one more extensions will be there, that is chromosome, which chromophores, which give color. Okay. Then about the flagella, bacterial cells may be motile or non-motile. If motile, that means if movement is possible, they have thin filamentous extension from their cell wall called flagella. So in the structure, cilia and flagella, the motile structures are the So bacteria show a range in the number and arrangement of flagella. It is composed of three parts: filament, hook, and basal body. See, these are flagellum, and this is an expanded view of flagellum. Okay, so which are the three parts? Filament, hook, and basal body. The filament is the longest portion and extends from cell surface to the outside. This is filament, the first part. Okay, then next one, hook. It's a curved sheath, connects filament to cell. Then basal body and curves flagellum into cell wall and membrane. Okay, so besides flagella, pili and fimbriae are also surface structures of the bacteria but do not play a role in motility. The pili are elongated tubular structures made of special protein, whereas the fimbriae are small bristle-like fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to rocks and streams and also to the host tissues. But for the movement, they require flagella. Clear, children? Then about the ribosomes. See, here you can see ribosomes, isn't it? So let's discuss about ribosomes. We know that both prokaryote and eukaryotes are having ribosomes. Now we are discussing about prokaryotic ribosomes. It is associated with plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size and are made of two subunits. See, here you can see two subunits. The 50S subunit, this is the largest subunit, and 30S subunit. And together they form 70S subunits. Children, please don't add it, okay? So the together it is known as 70S subunit. The large one 50S and smallest one 30S subunit. Okay? Children, what is the function of ribosome? Yes, protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes. The ribosome of a polysome translates the mRNA into protein. Children, in class 12, let's discuss more about the protein synthesis. Okay? Then about inclusion bodies. See, these are inclusion bodies. Reserved material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. These are not bound by any membrane system and lie free in cytoplasm. Example, phosphate granules, synophysin granules, and glycogen granules. Gas vacuoles are found in blue, green, and purple and green photosynthetic bacteria. See, these are inclusion bodies. Okay, here you can see pili. Can you see? But this pili, they are elongated tubular structures made up of special protein, but they never used for the process of, that means for movement, it is not useful. For the movement, they require flagella. See, this is flagella. Clear structure? So structure is clear, children. So that's about prokaryotes. And next class, let's discuss detail about eukaryotic cell. I know in class 9 we had discussed about eukaryotic cell. All the cell organelles we had discussed, isn't it? The endoplasmic reticulum, then mitochondria, then Golgi apparatus, plasmids. Do you remember it? So next class, let's discuss in detail about eukaryotic cell and their organelles. Today you study prokaryotic cell. Okay, children? Thank you.